Welcome everyone to the SNN Network Australia virtual event. I'd like to introduce you to our next presenter. Presenting on behalf of Prescient Therapeutics, publicly traded company, the symbol is PTX on the ASX. We have Stephen Yatomi Clark, CEO and Managing Director. Stephen, take it away.
Hi everyone, and many thanks for the opportunity to speak to you about Prescient Therapeutics. We're an oncology company based in Australia and the US. We're listed on the ASX, code is PTX. And we find ourselves not only on a wave, but out the front of a wave. And not only is this a very big wave, but we've got it almost to ourselves, which whilst many technology players become very, very crowded, we feel that we are in a unique position being ahead of this amazing wave. And I can't wait to talk to you about it a little longer. I'll just go through the disclaimer there and introduce the company by way of the investment highlights. We're an innovative oncology company. We're now focused not only just on targeted therapy, but on next generation CAR T. We've got a strong balance sheet, focused management. I'm not asking for money. We've got 2 years of cash, which is great. And we've got a team that punches really above our weight. So we are listed in Australia, as I said, code PTX. We've got not only Australian healthcare institutions on our register, we've got US life science focused funds on our register as well, which is unusual for a company of our size. I don't have time to talk in great length about our pipeline today, but I'll start with our foundational assets, which are our targeted therapies, PTX 100 and 200, which are in the clinic. Our knowledge of cellular pathways through our targeted therapy work led us to some cell therapy enhancement projects that we have underway and really opened the door for us to really some innovative targets and tools in CAR T, and that's led us to this amazing platform, which I will talk about as well. We also have a couple of COVID-19 programs going on, but I want to remain focused for this purpose on our oncology assets. And so I will start with our foundational assets, PTX 100 and 200, which are targeted therapies. And PTX 100 is licensed from Yale University. You'll find throughout the presentation we have a very simple business model, I guess. We license from the best and we work with the best. This one is licensed from Yale, and it's a first-in-class inhibitor of the RAS pathway. Now, for many of you know that following the oncology space, RAS is a very, very sought-after target with hitherto very little success. And this is currently in the clinic now and exhibiting clinical activity. Now, so companies like Amgen and Mirati, they've had a lot of headlines over the last 12 months or more on a very, very specific type of RAS mutation, G12C, and now some of them coming up with others. We believe our mechanism of action looking downstream of RAS captures many, many more RAS variants. So we believe we can capture what they cannot, which is going to be very important in the clinic when people are trying to target more than one RAS mutation, because typically patients who have a RAS mutation have more than one. And part of this downstream activity, without burdening you with terribly much knowledge about the molecule all at once, it inhibits this protein called Rho as part of this pathway. Now, Rho has become a target of interest in its own right. And what is interesting is that we've got the only drug in the world targeting Rho, which is very important in various types of hematological malignancies in particular. So it's a really nice sweet spot that we find ourselves in. So we have completed a previous phase 1 study done at Penn in Indiana, and we're now doing a basket trial, a phase 1B basket trial of solid tumours and hematological malignancies here in Australia. And what's interesting is, even though we're not powering this for efficacy, we are seeing a clinical signal already in the first cohort, which we did not expect to see. Some of these patients have failed up to 7 prior lines of therapy and are seeing responses that these patients have never received on any other therapy in their lives. So to get PRs and stable diseases, and now at the first cohort, at the second cohort, an unconfirmed partial response as well, is something that we did not expect to receive. So That is very encouraging indeed. PTX-200 is a targeted therapy for a different type of molecule called AKT, where it is a very novel, has a novel mechanism of action. It is not a kinase inhibitor. It is, in effect, a pH domain inhibitor. And we've established clinical proof of concept over a number of studies. I'll only focus on one for the purposes of this presentation, but previously we've shown a 91% overall response rate in a phase 2A HER2 study, HER2 negative breast cancer study. Those responses are durable. We have a companion biomarker. And also in ovarian cancer, we had 80% disease control in a phase 1B study before we even reached the optimal dose. So across a number of different studies, we've achieved clinical proof of concept. But in the case of AML, in relapse and refractory AML, this is really a cancer that gives other cancers a bad name. It is an awful disease, and once people are relapsed or refractory, it is not unusual for their life expectancy to be 4 to 6 months. Nevertheless, we're building on our successful phase 1 data at MD Anderson and the Moffitt Cancer Center, and now adding this to the standard of care, which is a chemotherapy agent called cytarabine, and we've already had 3 complete responses so far. Patients who were refractory and didn't have long to live that are now in remission and have gone on to bone marrow transplant, which is fantastic. And this drug has orphan drug designation by the FDA. So with 2 drugs in the clinic, they're producing data right now. Any one of them we think is going to be highly differentiated and be a company maker. But I'd like to now change gears for a moment and move on to cell therapy. And for those that aren't aware of CAR-T, it is an amazing new treatment approach that was pioneered by UPenn a few years ago. And it really is, some observers have called it a turning point in the history of human medicine, where we're seeing response rates that we've never, ever seen before. In fact, we've never seen anything approaching some of these amazing response rates. So it really is a massive paradigm change. And I'm aware that there's a blended audience here, and some of you are not immunologists or scientists, so I will just touch on what this means. So let's just backtrack a little bit. Firstly, what are T cells? And they're the soldiers of our immune system. They are designed to identify and remove foreign invaders. And these can be virus-infected cells. They could even be cancer cells. And it's all on the premise that these little arms on these T cells called receptors are able to identify things on the surface of these infected cells called antigens, and that identify these little structural proteins sticking out from the cell. And like a lock and key, these receptors are able to snap onto and recognize these antigens, and when they do so, they tear them apart. And is very, very effective at fighting foreign invaders. Now you're probably asking, well, why aren't they any good at removing cancer cells? Well, this is the thing, they are. And you may never know it, but if there's a cancerous cell pop up in your body, the tumor is very good at recognizing that and removing it so long as it can recognize those antigens on its surface. So it's like autocorrect. You wouldn't even know you've made a spelling error and it's corrected it for you. The problem is, those patients who end up presenting to the doctor with cancer have done so because these tricky cancer cells have changed these proteins on its surface. And now the lock does not fit the key anymore, or the key does not fit the lock anymore, and so they're incapable of removing them. So how do we overcome this? Well, it's very simple. We put a new receptor on these T cells. And when you put a new receptor on these T cells, the result is called a CAR T cell. They are now able to bind to these cells and tear them apart. Here's how the process works. A cancer patient will go to the doctor, blood is collected from that patient, and the T cells are removed from the blood. And remember, these T cells are currently not binding to these cancer cells. They don't recognize them. It's like a policeman walking down the street bumping into criminals without knowing that they're criminals. And outside of the body, we genetically engineer these new receptors on it that are now able to recognize these cells, and millions of these are grown up and infused back into the cancer patient. So the cancer patient is getting a turbocharged version, an enhanced version of their own immune cells. And this is not science fiction. As I mentioned, whereas other T cells, normal unmodified T cells, are unable to bind to the cancer cells, these ones can, and they latch onto it, and like any foreign invader, it will tear them apart. In fact, a single T cell can tear up to 1,000 cancer cells apart. So there is nothing so potent against cancer as your own immune system if we can direct it against it. So it's very important. It's the ultimate cancer killer. Yes, and it's not science fiction. This is earning real dollars for companies today. This is pioneered by Penn, and Novartis was the first to have a cell therapy approved. That little bag of cells you see there on the right is $500,000, which is a lot of money. Why? Because they work. And it is now spawned a multi-billion-dollar market, and it's only growing, even in the face of competition for this one particular target. So it really is a paradigm-changing technology that is growing very, very quickly. I'll skip over our cell therapy enhancement programs. These are designed to enhance current generation CAR-T programs. We've got 2 collaborations underway right now, which are designed to be almost like the Intel chip inside other people's computer, or the Dolby capabilities in everyone else's speaker system, and we'll be able to speak more about those down the track. I really want to talk to you about OmniCAR, and this is a next generation platform that is going to create next generation CAR-T problems, because like any new treatment paradigm, like any new technology, version 1 is going to be as bad as it gets, and that's where we find ourselves with CAR-T. How many of you remember VHS tapes? They changed the game when it came to watching movies. You didn't have to go to the cinema anymore and buy popcorn and sit down. You could watch them at home and invite your friends, stop and start when you like. But how many people have a VHS tape now? And that's where we find ourselves with CAR-T, which is great news for patients. As wonderful as CAR-T is, it's as bad as it's going to get. And these are the current challenges, the key challenges confronting CAR-T. The time and cost of a bespoke treatment, safety and control go hand in hand, because these are living cells that you infuse and you've got no control once you inject them. And targets and escape, a key chink in the armour of CAR-T is what happens if that tumour mutates again. And the people who pioneered CAR-T saw these challenges and invented this amazing platform that I'm about to describe to you. We're honoured to have licensed this foundational platform from the University of Pennsylvania. It was a deliberate collaboration between their cell biologists and their protein engineers to solve some of these problems. And here's how it works. It's a modular CAR-T. So there on the right, if you can skip ahead to the right, that's what a normal CAR-T looks like. It is created all at once, from the very start. That's what's infused. OmniCAR is different. It is modular. So step 1, a CAR-T has got basically all the signaling domain and, if you like, one half of the Velcro sticking out of it. And whilst this T cell is alive and ready to go, it is at this stage inactive until step 2, when you then separately inject the binding part of the CAR-T cell with the corresponding bit of Velcro. And inside the patient, these bind together and form an armed CAR-T cell. Now, this might seem very elegant and obvious even, but so often we see the most effective solutions are indeed so elegant as to be almost simple and obvious, and that's what happens here. And if you remember nothing else from this presentation, please look at this slide. And here on the left, if T cells are soldiers fighting cancer in the in the body, think of this conventional current generation CAR-T. Yes, they're a soldier, but they've only got one map and one weapon. They can only hit one target. They are incapable of being redirected. And most disturbingly, you can't control them or communicate with them once they're deployed in the field. So that's current generation CAR-T. And you look at OmniCAR on the right, which you can arm with any weapon, including several at once. You can give them any map for multiple deployments. And in fact, you can direct them against many targets at once and any target you like. Importantly, there's full communication and control even mid-mission, and you can even send images back in real time. Remember this slide because, if nothing else, this shows you how this is the generational change. And whilst we've got VHS tapes on the left and we've got Netflix there on the right, it is we believe this is going to be really, really important in solving these problems that are faced with current generation CAR T and bring them to more people. I won't go into these. You can see there's lots of crosses there against some of the challenges facing conventional CAR T and how OmniCAR is going to solve them. I'm just going to touch on a few. Well, one of them is safety and control. At the moment there on the left, there is no way of controlling these once you infuse them. You don't want to give so much that they're toxic. You don't want to give so little they don't work. You have to guess the Goldilocks amount. And the dose that you give is the dose that you give, and there's no control. By contrast, you can see with OmniCAR, we can actually titrate up the activity just by giving increasing amounts of this binder, just like a doctor does now with any other drug. You can escalate the amount that you administer to the patient, and this is no different. So it's like remote control for these cells. Once they're inside your body, you can control their activity up and up and up, if you like, to a dose that is going to work, but is not so toxic. But even if there happens to be a toxicity threshold breached, at the moment there's nothing you can do to even receive these life-changing therapies. These patients have to be in an intensive care unit just to receive the therapy, because that enables people to treat the symptoms. That's all they can do, the doctors, is treat the symptoms, and it has actually killed some patients. It's very dangerous. We're learning to control these a little bit better, but there's still massive safety issues. With this, you can see we can titrate it up safely, but even if there was a toxicity issue that was breached, then you can switch it off by simply stopping administering the binder. The therapy will switch off, but they remain viable and ready to go. So if the doctor wanted to switch them back on, you don't need a whole bunch of new cells. All you need to do is start re-injecting the binder. So you can switch it on, you can switch it off, you can switch it on, you can switch it off. So this gives the doctors the control that they've been craving for this type of therapy, which we've not been able to see before. As I mentioned a little earlier on, one of the key chinks in the armour of CAR T is, well, what happens if the tumour mutates? And all of a sudden the T cell has lost its nose again. It's lost its ability to sniff out the cancer. Well, with this, you don't need a new cell product. All you need to do is switch out the binder from one to the other, and all of a sudden you've redirected that one T cell to follow the cancer so that it cannot escape. So this is going to be important in diseases that are progressing very rapidly, and this very slide is something that companies are trying to address with CAR T in the relapse setting. This enables you to do it with the one cell product. But just as important as targeting multiple antigens sequentially is the ability to target them simultaneously. Now, this is going to be especially true in solid tumours when there's a lot of different antigens on the surface and it's tricky to sort one from the other. This sort of plug and play concept is going to be super important in being able to make better use of our knowledge of the antigens that we have. It's going to create a more upfront, robust response. And instead of addressing this tumour escape, it's going to prevent it. So this is going to be really important in the fight against solid tumours, which CAR T is yet to conquer. And amongst all of the data, I'll just share one slide with you, which is mice with ovarian cancer. And you can see that basically the upshot of this is the more binder you give, the more tumour you kill, and the longer the subjects live. So this is basically combining the unparalleled cancer-killing properties of a T cell, but you're giving it the control and the predictability of a pharmacological agent, of a drug. So you're combining the best of both worlds with OmniCAR. So when we've got a platform that is capable of doing almost anything in the cell therapy space, what do you do with it? Well, we're currently at the end of a strategic review to cherry pick which indications we're going to be pursuing in-house on our own balance sheet. It's probably going to be not one, it's probably going to be two now. But more importantly, we're going to be lighting as many fires as possible with anyone else in the field. If anyone else is in CAR T and they want to move their programs to a next generation one. They want to make theirs safer. They want to make theirs flexible and redirectable. They can be talking to us. Moreover, if there are companies out there with antibodies and biological programs that aren't even in CAR T, guess what? They can take their products off the shelf, even if they're not even in the market. They can lifecycle manage those by turning them into CAR T programs with our platform. So I think the field is littered with people who are sitting on the fence thinking that the second mouse will get the cheese, and I think that is largely true. And for those sitting on the fence with their own programs, this is a way of not only getting them into the field with their current assets, but leapfrogging people to have next generation CAR T therapies. So this is the response we're getting from this is astounding in the few short months that we've had it. Some of the deal activity has been well documented, and if you look at the dates of these, a couple of these are all on first generation types of CAR Ts, and they're for eye-watering sums. The only thing I would mention here is that one of the founding scientific appointments at Juno, Alan Ebens, has recently joined our board because of he so believes in this. He says, Stephen, this is what we're trying to do at Juno. This is what you've done here with what OmniCAR is capable of doing is the holy grail. So this was sort of first generation deal activity, if you like, but as evidence as to where we're going and what is happening in the field, Astellas purchased Xiphos. Xiphos is was a company developing their own modular system. For reasons I won't go into, we believe OmniCAR is best in class, has got many advantages over this. I won't go into that right now, but we think they're significant. They were at a similar stage of development. Astellas, who realized the same challenges that I mentioned before in their cell therapy programs, knew that modularity was their solution, and they bought a Xiphos on Boxing Day last year for $665 million. So I think we believe that this is maybe the first of the next generation deals, and we find ourselves at the front of that pack, which is wonderful. So there we are. So what else can OmniCAR do? Well, it's going to be applicable not only to hematological cancers, blood cancers, but solid. It has applications even beyond oncology into diagnostics and autoimmune diseases. These are areas that some big companies are telling us now. They say, Stephen, we really think CAR T can benefit some of these autoimmune diseases, but we cannot justify the safety profile. We cannot justify the risk reward with current generation ones. If you can give us one that is safe and we can switch off, all of a sudden we're listening. Well, that's what this enables us to do. And importantly, we are totally agnostic on the type of cancer, on the target, and even on the cell type. People are talking about, sort of their own, autologous T cells versus off-the-shelf T cells. I couldn't care less. People are arguing over T cells versus NK cells. I couldn't care less. This can improve any cell therapy approach due to its modularity. So incredibly exciting opportunities in front of us, and absolutely the right time for it. These recent conferences have confirmed that for me. So we find ourselves towards the end of the presentation. I know that's a lot to take in, but if I can leave you with anything, it's that there are lots of levels of value here in Prescient. We punch above our weight. As I said, we license from the best and we work with the best. We've got our targeted therapy portfolio. They're in the clinic. They're spitting out data now. Any of those is a highly differentiated company maker. We have our cell therapy enhancement programs, which build on that and has direct relevance to people on the manufacturing and adjuvant areas in cell therapy right now. We have OmniCAR, which is a game changer for the field, we believe, and is not only going to yield next generation products for us, but for anyone else, not only in CAR T, but in the biological field as well. And if that wasn't enough, we have some COVID-19 programs as well, which I leave that off to the side because we're not running those. We've given our assets to the Doherty Institute to run those through their screening program, and those are out. It says September, October, but that's been pushed out to November, which means it is imminent. So lots of activity, lots of news flow, and I thank you all for your time. Our code is PTX on the ASX, Australian US company, and we've got lots of cash, and we find ourselves in a situation where we're at the forefront of a really big arbitrage. So many thanks for your time. I look forward to your questions, and I look forward to welcoming you as a shareholder. Thank you.